welcome to another episode of the Iraqi Nutrition Podcast. As always, I'm your host, Juma Iraqi. Before we start today's episode, I just want to mention that this podcast is available on YouTube, but you can also find it on SoundCloud, Stitcher, and iTunes as well. So today's topic is muscle physiology, and I couldn't think of a better guest to invite to talk about the things that we're going to talk about today than Professor Darryn Willoughby. He's probably the most muscular professor you'll ever see, and probably one of the smartest when it comes to muscle hypertrophy as well. So Darryn, thank you so much for coming back on the channel and discussing this topic with me. I think this is the third time I've had you on, so that speaks in volume how much I actually appreciate bringing you back to this channel. So thank you so much.
You're very welcome. It's always a pleasure to be here. So and chatting with you, and and like you, we both are so passionate about helping to try to educate others about what we love so much. So
absolutely, and I know how busy your schedule is, so your time is really appreciated. So Darryn, for people that might not know who you are, could you give us a introduction about yourself?
Yeah, sure. I'm a professor of exercise physiology and nutritional biochemistry at Baylor University in Waco, Texas, here in the USA, and I also run a research laboratory, exercise and biochemical nutrition laboratory, where we do research on a number of different topics that involve either resistance exercise, resistance training, or perhaps some type of nutritional intervention, specifically in skeletal muscle, looking at aspects both at the molecular level, meaning gene expression level, and protein level, how it translates into protein, and perhaps then impacts muscle growth and muscle performance as well. My PhD is from Texas A&M University. It's in specializing in exercise and muscle physiology, and subspecializing in nutritional biochemistry and molecular biology. And then my bachelor's and my master's degree were both in exercise science. And so, I also have a practical side to me. I'm a certified strength and conditioning specialist and a certified sport nutritionist. And so, I work on the side and consult with people, particularly relative to, nutrition and dietary strategy. And then I also do prep work for competitive physique athletes, whether it's, female bikini or figure or physique, or male physique or classic or standard bodybuilding. So right now I'm prepping, I believe, 6 people for upcoming shows. And so, and myself, I'm also a competitive bodybuilder still at the age of 55. I still compete. I love training. I love competing. And so, it's just something that is a passion of mine, and I love doing. And so I love being able to kind of practice what I preach, so to speak, to my students. They know that I'm in the gym every day, and what I'm telling them is not not something that I'm just making up. It's it's something that I that I live each day in my personal life as well, not just my professional life. So
awesome. I think the last thing that you mentioned there is really important to let people know, because a lot of people, when they look at research, they just imagine people in white lab coats that have never lifted a dumbbell in their lives, and that's not really true. Like, most of the people that I know in exercise science, researchers, are either currently competing in some sort of sport, or they've been formerly active themselves as competitors. But before we jump into the question, what are your thoughts on if you look at professional bodybuilding now compared to what it was maybe 20 years ago, what are your thoughts on the development that we've been seeing? Because now it seems like there's more avenues that you can select. Like, in the past it was only bodybuilding, but now you have like bikini and men's physique, and some people might say that it's been positive that we have several avenues that you can select from. But to me it seems like bodybuilding on the professional level is slowly dying in some way.
That's a good point, and I would tend to agree from the standpoint that, well, we basically already know that in the IFBB, for instance, women's bodybuilding is basically gone. So it's been for the most part replaced professionally with women's physique, and so I'm also a competitor and judge in the Global Bodybuilding Organization. It's a newly founded organization. It's about 3 years old, and so it's growing and booming quite well. But but we're not trying to necessarily compete with anyone else. We just are who we are. But the point I was going to bring up is that when we go to shows now, we see just most of the competitors at the shows are, and for the other on the. I would say it's also very similar with the NPC as well, and some of the other organizations, is that women's bikini, women's figure is just exploded. Men's physique has just exploded. That's where most of the competitors in a given show, those are the classes that they're competing in, and we're beginning to see less and less and less and less in standard bodybuilding. And even classic bodybuilding is actually growing somewhat as well. Mm-hmm. And so, it's interesting. And I'm all for giving athletes, other avenues in which to compete in. Not everybody wants to be a bodybuilder per se, but, if they have a competitive spirit in them and, when they're going to the gym each day and they'd like to showcase, their efforts, then why not, But, in terms of kind of what's caused the decline, particularly in standard bodybuilding, it's hard to say. I would say probably for women, just from the standpoint of probably, at least in the United States, I guess maybe kind of a public outcry, if you will, or a lack of tolerance anymore for the physiques that were being displayed in standard bodybuilding, and in some cases the amount of masculinization that was being seen in some of these female competitors, and don't get me wrong. I respect, you've got a female standard bodybuilder. I know what goes into that. And so I respect what she does to the nth degree, but, but that's me. Mm-hmm. So I would say that, at least the public had grown a bit, I guess, tired, concerned, whatever the term you might want to say, with competitors that and I've heard people use this term, competitors that look like men wearing bikinis on mm-hmm. stage, mm-hmm. And I think it comes from a standpoint of a number of things, but probably women shouldn't necessarily look like that, and as a result, we're not necessarily going to support that. So that's probably one of the reasons, or at least one of the several reasons, that women's bodybuilding is kind of taking a big hit. Men's bodybuilding, I don't know. Maybe people are kind of growing a bit intolerant to seeing, the freak show. Bodybuilding in and of itself, particularly standard bodybuilding, is kind of has a cult following anyway. There are those of us that appreciate it for what it is, and we like to see the kind of weird or strange or peculiar, and so when you see, particularly at the IFBB level, like on the Olympia stage, some of these guys are just unbelievably huge and dense and thick, and then sliced on top of it in terms of their conditioning. That's pretty freakish, Mm-hmm. So, I don't know. It's hard to say. There are probably a number of factors, but I think probably also a number of competitors have migrated away from that into maybe more of a classic bodybuilding approach or maybe a physique approach because, maybe they don't want that much muscle, or they don't want to work for that much muscle, or whatever the case may be. Or perhaps maybe they don't feel like that they have to submit themselves to the use of pharmacopoeia to be able to attain that level of muscle. And so, I think it's multifaceted, but and there are many reasons why. But nevertheless, it standard bodybuilding has, and even with the men, has definitely taken a hit also. So it'll be interesting to see where it all goes in the next 5 or 6 years. So.
Yeah, maybe it changes now, since now you also have a new Mr. Olympia, so it might be different criteria they will have for the optimal physique. So maybe they're getting away from the mass monsters. So we'll have to wait and see.
Well, and that's a very good point, because I think with Sean Roden, I mean, obviously he brings a ton of mass, but he also brings it on a very aesthetic, more visually appealing physique. His symmetry is great. He's got the nice tiny waist. He's not dealing with the bubble belly and that most of the other competitors are. And he's just, he's big, he's massive, he's thick. But as I said, he has a beautiful, aesthetically pleasing physique. So your point is a very good one. I think, and he's kind of got a bit more of what I call a retro physique, if you will. He kind of back, 15, 20 years ago or so, like that, even though he's thicker and denser than a lot of the physiques back then, but still his lines are very, very similar to that. So it'll be interesting to see what happens, now that we kind of have a changing of the guard, and if that's going to kind of now be the criteria that the judges are going to kind of be more concerned about. And and then that being said, if once the competitors have a good idea of what the criteria they feel the judges are looking for, then in their training they start trying to strive to basically to obtain that that type of physique. So I agree. I think you're totally correct about that.
Yeah, we'll just have to wait and see, but that's my hunch on it. So anyways, let's get into the question. I really appreciate your take on that, by the way. It was interesting to hear your take. So I think we'll have similar thoughts on what we're observing in the industry. So let's start off with explaining how does a muscle actually grow.
Okay. Well, there are a number of factors that will impact the process of muscle hypertrophy. I guess we can start off by just generally describing, that, I guess two factors will impact this, what we call more of local factors and more systemic factors. Systemic factors meaning that factors that arrive from the system, from the body. And these primarily would be hormones, growth factors, testosterone, growth hormone, insulin-like growth factor 1, for instance, in terms of, anabolism, myostatin being down-regulated with exercise and training would be another, since it is a negative regulator of muscle mass. So, you can talk about those types of scenarios where, those hormones will interact with muscle, bind with the receptors, and then they activate signaling pathways in muscle to actually increase muscle-specific gene expression and also muscle protein synthesis. All right. And then local factors being those that simply are just governed by changes in the architecture of the muscle itself associated with training. So the term that it's typically used is called mechanotransduction. Once the muscle fiber is being shortened and generating tension, and then it's being lengthened and it's being stretched, the muscle membrane ends up activating various signaling pathways in muscle as well that can also play a role in increasing gene expression and muscle protein synthesis as well. So, we know that muscle can be impacted positively simply by virtue of this mechanotransduction, independent of some of these systemic growth factors that we've mentioned. And there's data showing that the mechanotransduction mechanism is for the most part much more powerful than the growth factors that we see. And for instance, if a person is not doing resistance training and they're administering themselves anabolic steroids, they're going to have, for instance, or even growth hormone, or they're going to have a modest effect at best. That means that what's the primary driving stimulus for the anabolic response in muscle to grow? All right, it's mechanotransduction, meaning it's the process of the muscle put under mechanical stress. So since it's hard to exclude the systemic impact as well, meaning that during exercise the body will release some of these different hormones and growth factors just as a part of training, and even during the recovery period as well, is that we know that muscle grows primarily and collectively due to the culmination of both of these of this mechanical events, if you will, and the conjunctive influence of the growth factors as well. So.
Great, that was a great explanation. I have a follow-up question to that. Now, we know that when you age, it seems to have it's much harder to build muscle as you age. What is actually happening as we age on the cellular level?
Well, that's a good question, and that could be due to a number of things. One is that, and the other thing that I didn't mention in the previous question that's probably a bit more of a indirect scenario that impacts muscle growth, and that's obviously nutrition. We have to provide, obviously, the body and the cells with fuel so that they can, the cells can make ATP and create energy to substantiate muscle contraction. So that obviously mechanotransduction can occur. Mm-hmm. So, I would say in some cases with aging, it could, nutritionally speaking, it could be from the standpoint that perhaps, as we age, maybe our nutritional profile or our dietary approach may not be optimal. It may be suboptimal. We know many times as people age that's very, very, very common. Mm-hmm. A lot of times we see that people have a tendency to undereat as they age. And, the other thing potentially could be, but there's some data to nutrition-wise to suggest that as we age, there's a consequence known as leucine resistance, that meaning that as we, the muscle becomes more resistant to the impact of leucine in being able to stimulate muscle protein synthesis. And so, if that being said, that would imply that as people grow older, they need to ensure that they're taking in adequate amounts of protein over and above what they may have needed prior due to the leucine resistance. And if they're not doing that from the standpoint of either under eating on a routine basis or not eating enough protein, this could obviously impact the muscle's ability to grow. The other is that we know that as we age, we begin to reduce the number of satellite cells that we have in us, in our muscles throughout our body. When we're young, we have a large number of those, but if we're active throughout our lifetime, and we continue to utilize satellite cells and call them into activation, that at some point later in life, then it's not presumptuous, and there are actually data that indicate this, that it appears that satellite cell number begins to reduce, or becomes reduced. And so that being said, then the muscle can lose its ability to regenerate itself in the face of resistance exercise. So that could also be another issue as well. But the whole process of aging and muscle function and aging in general is so unbelievably multifaceted. So that would only be just a couple of items that would be most likely. I mean, we know that as we age, particularly more advanced age, we start to upregulate telomerase activity, which is an enzyme that begins to degrade telomeres on the chromosomes. And so when that happens, then we begin to lose, genetic potential or genetic power, being able to drive gene expression, because DNA starts to become degraded. So, that would be something that would be a bit more of that happens, but in terms of your question, that would be something that would be a bit more of a stretch. The ones that I feel would definitely be kind of on the front line in terms of the answer to my question would be nutritional, would be nutritional aspects, and also this reduced ability to regenerate muscle due to satellite cell activity as well. And then the other is that we know that neurologically, as we age, neurons begin to start to degrading a bit. There's some data showing that there's a protein called agrin that's associated with the neuromuscular junction, which is the juncture point between motor nerves and muscle fibers. And that as we age, this agrin starts to become degraded, and so there's a marker that's used for other aspects as well with aging, but particularly in muscle, it's a marker that's used to indicate degradation in neuromuscular junction, and that is something's called the C-terminal fragment of agrin, because agrin begins to be degraded at the C-terminal, and then that ends up finding itself in the bloodstream. So people are using that and being able to assess that, and we're seeing that as people age, the levels of that biomarker are higher in older people compared to their younger counterparts. So again, if that's indicative of a reduction in neuromuscular transmission, then that means that would impact the muscle contraction as well, and the muscle's ability to perform at somewhat of an optimal level to be able to generate a proliferative, hypertrophic response. So
interesting. I love doing this podcast because I always learn something new when I have great guests on. You mentioned something about satellite cells. So for listeners that might not know what that is and how that fits in the whole process of skeletal muscle remodeling, could you please explain a bit about that?
Yeah, satellite cells are basically just muscle stem cells. So once we are once we're conceived and develop and in utero, and once we're born, we have these stem cells, if you will, that have been committed to the muscle lineage, but they've never proliferated into adult, fully differentiated muscle fibers. So they lie below the basement membrane, at the sarcolemma, and so they're kind of along the membrane of the muscle, and they're just there in a state of quiescence, in a state of silence, if you will. And so, but we know that under different types of muscle stress, that these satellite cells can be activated, and once they're activated, then they can differentiate and proliferate and form what are known as myotubes. And they don't form new muscle fibers. They form what are called myotubes, which have no contractile activity, but they're multinucleated, like our skeletal muscle fibers are as well. Our adult fully differentiated muscle fibers are multinucleated. So with, for instance, muscle stress, mechanotransduction, muscle damage that occurs with resistance exercise or any exercise, but particularly heavier resistance exercise, as we all know, causes muscle damage. And so this muscle damage will actually stimulate the activity of these satellite cells, and this is all part of the process of muscle regeneration, muscle recovery. And so the satellite cells, as I said earlier, end up forming myotubes. Then they fuse with the muscle fiber, and then they donate their nuclei to the adult muscle fibers, to the fully differentiated fibers in which these satellite cells were associated with. So now what happens is that muscle fiber now has even more nuclei, which means that it has even more DNA now within that. Those DNA contain genes, and those are genes that will be expressed during expressed during resistance exercise, and thereby have the ability to translate into new proteins, to synthesize new proteins. So so when we talk about damaging muscle, some of the protein in muscle is damaged beyond repair. Some protein is slightly damaged, and then the heat shock protein system, particularly heat shock protein 72 is a molecular chaperone, can end up helping to repair some of these partially damaged proteins. But a number of them are damaged beyond repair and will simply be completely proteolyzed, and then the cell will rid itself of them. So that means that the damaged muscle has to replace those damaged proteins with new protein. And so it has the ability to do that quite effectively by satellite cells being activated, so that it can augment, if you will, the muscle's ability to stimulate protein synthesis. So, and this is another thing. Over time, this is a very important process in terms of muscle hypertrophy, because each time we go into train, we are mechanotransductively activating the hypertrophy process. But if we're training intensely enough, then we're damaging muscle each time we go in. This is the whole concept behind progressive resistance exercise and the overload principle. Again, that's why we don't want to go in the gym, every day, day in and day out, and lift the same amount of weight. After your muscle is going to hypertrophy up to a point, and then that's it. It gets no larger after that. Mm-hmm. So we want to consistently, intensely try to damage muscle in a way that we're going to call into play the activation of satellite cells to give us a robust amount of regenerative power in muscle to help provide a hypertrophic response.
Interesting. If I recall correctly, I also think there was a study that you were involved in looking at creatine and what actually affects these satellite cells. Is that correct? Isn't it?
Well, we did a study, and we didn't actually directly assess satellite cell number, satellite cell function. What we did is we assessed the concentration of proteins, the myogenic regulatory factors, MyoD, myogenin, MRF4, that are involved in the process of satellite cell activation, differentiation, and proliferation. And so those guys come into play at those different levels or different stages to be able to ensure that once a satellite cell is activated, it can actually wind up forming a myotube. And so we did a study, and it actually was kind of based on a study that we saw that was done in the couple of years, published a couple of years before ours, by Peter Hespel, and it was a very, very nice study looking at creatine supplementation and in terms of training in a rehabilitation setting. And so his group saw somewhat of a similar response as to what we saw, is that the creatine supplementation group provided much more of a kind of a robust platform for the expression of these myogenic regulatory factors. Now, there was a very, very nice, elaborate study that was published in, I believe it was 2007, by Olson's group, and they did 16 weeks of resistance training. They gave either a control scenario, or they gave protein supplement, or they gave creatine supplementation. And they actually measured satellite cell number and satellite cell activity, so they actually took a more direct approach at measuring satellite cells and the activity of those, and what they found is that protein supplementation was better than control, but creatine supplementation was better than protein and control, and it caused a very, very nice, significant impact in satellite cell activation. So it's been suggested that, and we don't know the mechanism, but it's been suggested that creatine may play a role as somewhat of a cofactor, if you will, in helping some of these myogenic regulatory factors to be able to be involved or play a role in the process of satellite cell activation. Again, no one knows the mechanism, but there's enough data from several studies, mine being one of them, but particularly with the Olson study, that was definitely, certainly much more of a direct approach, in showing that. And in the Olson study, they also showed that there was a greater improvement in performance in force production in the creatine supplemented group as well. So they had a nice practical piece to that study as well. So, but again, back to that, it's kind of hypothesized, if you will, that creatine seems to have some type of a cofactor role in being able to work with those myogenic regulatory factors to activate satellite cells. So at this point, that's the best we know, but we know that there's some type of relationship between creatine supplementation and satellite cells.
Interesting. Now let's move on to, you often hear the term androgen receptors. So could we explain what androgen receptors are and what effect they have on potentially growing muscle or limiting the ability to grow muscle?
Sure. So, in a number of cells in the body, but specifically in muscle, within the muscle cell are androgen receptors, and so hormones that are lipophilic, meaning that they're fat soluble, like all steroid hormones are, testosterone, estradiol, cortisol, for instance. These, unlike peptide hormones or amine hormones like norepinephrine but peptide hormones like insulin and growth hormone and IGF-1, they bind to an extracellular, or what we call a transmembrane receptor. So they bind outside of the cell and dock, and then they transduce their effect inside the cell. With testosterone, it's different from the standpoint that based on its lipophilicity, it's able to find its way across the cell membrane, and then inside the cell is the androgen receptor. And so the testosterone will interact with the androgen receptor inside the cell, in the sarcoplasm. In an unbound state, meaning that the androgen receptor is not bound to testosterone, it is inactive, and it's held that way because of its association with the small and larger heat shock proteins, 40, 90, 70, and then once testosterone binds to its androgen receptor, then that inhibition is relieved from those heat shock proteins, and then the androgen receptor becomes activated. Now, the other thing is that, for instance, is that when testosterone comes into the cell, then there's an enzyme called 5-alpha reductase that can also convert testosterone into dihydrotestosterone, or DHT, which has a greater binding affinity with its receptor than does testosterone. So some will bind to testosterone, some testosterone will be converted to DHT, some will not. We did a study and published a study in the journal Steroids about 3 years ago, the androgen receptors, and we actually measured and assessed, because it'd been stated in the literature that there was very little to any 5-alpha reductase activity in skeletal muscle. Well, we showed that wasn't necessarily the case. It was about a 50-50 when we looked at muscle testosterone concentration and muscle DHT concentration. So and the only way that can happen is if you have a fairly active 5-alpha reductase enzyme. So anyway, once either test or DHT binds to the receptor, either one, they'll activate the receptor. Now, the receptor, which is a protein, it has what's called a DNA binding domain associated with it in its sequence. And once the androgen binds, then that active complex, that hormone receptor complex, becomes, as I said, activated, and it becomes a transcription factor. So it will translocate from the sarcoplasm into the nucleus, and then by virtue of the DNA binding domain of the receptor, it will bind or dock on a region in the regulatory promoter area of various muscle-specific genes called an androgen response element. And once it docks there, it will then increase the expression of various muscle-specific genes, and many of those genes are going to translate in code for myofibrillar proteins, so like actin and myosin and troponin and tropomyosin and a number of other proteins within the sarcomere that are going to be involved in substantiating muscle contraction, and over time, as those proteins continue to proliferate, then they're going to provide a hypertrophic response to muscle. So in general, I mean, that's the androgen receptor. That's how it works. Obviously it's quite a bit more complicated than that, but in a general sense, that's how androgens work in conjunction with the receptor to instigate a response in muscle. So,
wow, is this going to be on the test tomorrow?
I don't know. You might want to inform your listeners. Okay, pop quiz tomorrow.
Pop quiz tomorrow. Yeah.
Awesome. Hey, believe me, my students get tired of hearing this. They hear me talk about this stuff all the time. So sometimes it's like, okay, here, Dr. Willoughby, here he goes again, off on a tangent. So yeah, so it's kind of a common thing with them. But oh well.
Well, here you have almost 11,000 muscle heads on this channel, so I think they will I think they will appreciate it. But I think it's really interesting that we went into this, because my follow-up questions on the androgen receptors is, can you, like, is it genetically, like, can you have more androgen receptors in a specific muscle and less of it in another? And how would that potentially affect if you can grow muscles in that area or not? And are there anything that can affect an increase in androgen receptors or decrease in androgen receptors?
That's a good question. And and the interesting thing about human physiology and biochemistry is that, especially when you put exercise in the mix, and that is, this is something I tell my students all the time. Matter of fact, I mentioned it last night in my graduate neuromuscular exercise physiology class, is that, in terms of human physiology biochemistry, very seldom is anything absolute. It's always somewhat relative, because you have such a dynamic, ever-changing system. But to answer that question, I was going to say, in terms of somebody having more androgen receptor content, than another person, or perhaps more androgen receptors in one muscle compared to another muscle, that might factor in as to why somebody might be a hard gainer. They train their legs as much as the same intensity and volume as they train their upper body. Their upper body grows, but their legs don't, or vice versa. We all know people like that. So, it's hard to say exactly why that might be, but that could be a possibility. Because we see alterations in physiology that impact physiological outcomes all the time. So it's possible. It could instead perhaps be that it may not impact the androgen receptor. It may be something where somebody has a bit more of an overactive expression of myostatin, or something like that, where they're actually getting a negative regulatory impact that way. We know, for instance, that testosterone will actually downplay or silence the activity of myostatin, by the way. So, that's one of the other reasons, that the testosterone, particularly if given in, larger amounts, if somebody is self-administering it to themselves, provides a hypertrophic response, based on what we just talked about, but also because it has a silencing, inhibiting effect on myostatin. And it also, and that happens because it helps to upregulate MyoD. And then if we upregulate MyoD, we also know that MyoD is involved in satellite cell activation process as well. So testosterone can be impacting muscle hypertrophy through the, number one, upregulation and expression of muscle-specific genes, just pure and simple, based on that specific mechanism. Number two, in increasing the expression of and the concentration of MyoD, which itself is a transcription factor and can impact expression of muscle-specific genes. And the third is the fact that it can also silence myostatin as well. So, but back to your question, it's, I'm not specifically aware of any data, and it may be just because it may be that it's there, and I haven't come across it. I haven't come across it about the differences in androgen receptor content, but it's very possible. Yeah, I wouldn't discount that. So,
yeah, that's especially interesting what you're mentioning about, like, supraphysiological doses is decreasing activity of myostatin, and I want to get back to that, but Does
Well, and there's something else that I was going to, if I could. I
Yeah,
mean,
of
you
course.
mentioned you asked me this question and I overlooked it,
Mm.
and that you said, is there anything that could impact the concentration or the amount of androgen receptors? And so we know that, um, I mean, prolonged exposure to androgens above and beyond the normal physiological range will cause downregulation in the androgen receptor.
Mhm.
So we can decrease androgen receptor density content by downregulation, and that's again due to, I guess, sustained exposure to high amounts of androgen. So if somebody is administering themselves a pretty moderate to moderately high or very high dose of, you know, the higher the dose, the more the downregulation.
Mhm.
Because what happens is that over time those receptors become completely saturated, and there's still androgen there. So the body will downregulate, and it's not just with testosterone. The body does that with other receptors as well. We see that happening with different drugs that we take. That's why a lot of times, physicians will ask that people take, various drugs for whatever condition, and they'll ask them to cycle them, takes take this many for so long, and then change your dose, and so that they can try to keep somewhat of a robust receptor response, so that they can continue to get a sustained effect with those medications. You know so it's a dose response type of scenario,
Mhm.
particularly with testosterone and the androgen receptors. Um, but we usually don't see that, in somebody, if they're not administering, and they're just relying on their body's normal level of testosterone production. You're not really going to see that, because the body, based on negative feedback, will maintain a very constant level of circulating testosterone,
Mhm.
and as a result, so that we don't, physiologically, the body won't necessarily allow for androgen receptor saturation. So it's not something that we typically, under normal physiological circumstances, will find ourselves dealing with. So,
Interesting. Now I want to go back a bit and talk about myostatin that you mentioned.
okay.
Could we mention what it actually is and what role it plays in the amount of muscle or the lack of muscle mass you can actually build?
Yeah, myostatin is a protein it's expressed and synthesized in muscle, um, and then released from the muscle. It can work in an endocrine, autocrine, and paracrine fashion, meaning that endocrine is that, once in the blood, it can circulate and impact skeletal muscle and other parts of the body. Let's say, for instance, myostatin is being released from the pecs, and then or it can be released and impact adjacent muscle fibers, fibers that are next to it, or it can be released from a particular muscle fiber and then impact the muscle fiber in which it was released from. That would be called um, an autocrine scenario. Adjacent would be paracrine. But anyway, so it's a protein that's synthesized. There's a myostatin gene in muscle. It's expressed, and then that mRNA will translate into myostatin protein. But in the cell, that protein is in a kind of a what we call kind of an inactive state. It's in an immature state, and then as it's released from muscle, it ends up assuming a mature state in circulation, and then it has the ability to bind with its receptor on muscle membrane, called the activin 2B receptor. But we have other proteins in the blood that can actually inhibit myostatin from binding to that receptor. one, first and foremost, it's called follistatin. And then there's another protein that's similar to it called follistatin-like related gene. And then there's another called GASP1. But follistatin is certainly the protein that seems to have the most impending effect on its ability to block myostatin binding to its receptor. If myostatin does bind to its receptor, then it transduces a signaling pathway in muscle that does two primary things. One, it inhibits satellite cell activation. And two, it can upregulate a pathway in muscle that breaks down muscle. It's called the ubiquitin proteolytic pathway. And so what it does, it's, myostatin is regarded as a negative regulator of muscle mass. I mean, I've seen people theorize that in humans we have, under normal physiological circumstances, we have a physiological ceiling for muscle size, and that it's governed by myostatin. Not sure whether that's actually true or not, but I've seen reference to that. Of course, that, again, I have to preface normal physiological scenarios, because, again, with heavy doses of androgens, for instance, we know that we reduce myostatin activity. So we wouldn't consider heavy doses of androgens exogenously provided to be under normal physiological circumstances, So myostatin has the ability to inhibit muscle regeneration because it blunts the ability of satellite cells to be activated. And then the other is that it can play a role in proteolyzing or degrading intact muscle as well. So it can cause muscle atrophy that way. So, yeah, and it can have a pretty prolific response just based on a good amount of the animal data that I've seen. So where they were able to overexpress the myostatin gene, and they saw pretty dramatic impending outcomes on muscle atrophy. But then again, I think everybody's probably seen the pictures of where we are able to transgenically, knock out the myostatin gene in animal models, and then these guys are just huge. The mice that they've been able to do, the McFerrin's group, back when. And then there's studies where they were actually able to, instead of that, they were actually able to create transgenic models that overexpressed follistatin. So they were actually able to, where they were actually able to transgenically provide mice that didn't express the gene that would allow for the production of the activin 2B receptor. So there's no receptor there for the myostatin to bind to from the bloodstream. And so those mice got big and swole as well, because they were able to silence myostatin based on either knocking out the gene for myostatin or knocking out the gene that provided for the receptor for myostatin. So, so we know what it's capable of doing. In humans, I mean, there's really no way that we can control it or block for it. I mean, they've been working for a while on drug trials, particularly for Duchenne's muscular dystrophy, for a myostatin inhibiting drug, if you will. But at least in us, under anything that's FDA regulated or anything like that, then I've heard talk of outside of this country. I've heard of talk of drugs that have been developed over there that are myostatin inhibitors, and that and other things is why some of the some of the top IFBB pros have just, in like a year's time or year and a half, they've just gotten even freakier than they were before. So whether that has anything to do with that, not sure. But, the problem with somebody tinkering with myostatin inhibiting drugs, though, is that myostatin is also expressed in cardiac muscle. So, if you inhibit the activity of myostatin, then that means that cardiac muscle can also can hypertrophy as well, and that could result in cardiomyopathy or extremely enlarged heart, typical to what a lot of times what we see with congestive heart failure. And so, it's very possible somebody could or would succumb to heart failure at a relatively early age. So I don't know if that fully answered your question about myostatin, but if not, I'm more than happy to try to.
No, absolutely. I think it was exactly what we were looking for. And for the listeners, if you've ever seen a Belgian blue cow that's are really swole, it's not androgens that they're putting in their foods or injecting them with it. It's actually they have. But in that case, it's not tinkering, right? It's not. Nobody's tinkering with their.
No. That breed of cattle is known for having defunct myostatin genes. So that's just how they, some of them are born with a mutated myostatin gene, and so they get what they call double muscled. There's also a breed of dog that happens to quite a bit. It's called the whippet, and so it looks kind of like a greyhound, and they fairly often get that way as well. So but the Belgian blue, I mean, that's and you're right. There's no tinkering. That's just because they have, and you know what? There's been a couple of scenarios where. And there was a number of years ago, they found a boy that was born that way in Germany.
Yep.
He had a mutated myostatin gene, and so, I mean, he was just growing like crazy, and putting on muscle like crazy as well. So
he had calves at 9 months, I think.
I mean, you saw the same pictures. Yeah. He was. He was jacked at by a year old. So but I haven't seen any. I don't know. I haven't seen any pictures of him, because now he would probably be about what, maybe 10, 12 years old, something like that. So I don't know. It'd be interesting to see, but it's I don't know. It's probably, I'm sure the parents are trying to probably keeping him out of the limelight. I don't know, but there have been instances in humans where that's happened as well. So But again, what scares me either about that or somebody else who is giving themselves, if they are in fact able to get access to an actual myostatin inhibiting drug, my fear is that the impending effects that it could have on their heart and their cardiovascular function. That's pretty scary stuff.
Yeah, absolutely, and I think that's like if we were to find a drug that actually couldn't inhibit myostatin, it would be wonderful for the things that you mentioned with muscle atrophy in elderly, for example, and we could use that for treatment. But you also run into the risk of people actually abusing the drug and not really thinking about the long-term consequences, based on what you're mentioning with an effect it has on the cardiovascular system.
Yeah, and if at least if that being the case, I mean, they would likely, in terms of the drug that we're, for instance, assumably here in the States with the FDA approval, they would kind of determine an effective but dose that's but yet that's one that's safe at the same time, and to be able to provide and get a positive impact in skeletal muscle, perhaps while minimizing any impact on the heart and the cardiovascular system as well. So, but there's a potential way to look at it is if somebody were older and they were being given this drug perhaps to help with combat sarcopenia, if they were also having struggle with perhaps weakened cardiovascular function, this could potential maybe help strengthen the heart a bit, at least with its contractile efforts. But again, that's being very presumptuous, but it's certainly not out of the question. But nevertheless, I think that we're going to end up finding out more and more and more here in the next few years about myostatin and in terms of its use in humans, because I'm sure that at some point it will become available, and people that it wasn't necessarily designed for in terms of its creation, like bodybuilders, will start experimenting with it. And I suspect that they already have, based on some rumors that I've heard, and in the changes in physiques that I've seen in a relatively quick turnaround. So, but don't know that for sure. So we'll that remains to be seen. So, but myostatin is a very unique protein in terms of its capability to impact muscle the way that it does. And so, it's and not only that, how it's regulated as well. And because of that, we know that there are various ways that we can experimentally perhaps impact its effect. I've heard of people here in the States that are getting access to some of these companies where they can buy follistatin and are actually injecting follistatin with the hopes of being able to inhibit myostatin. So, but but even then, that scares me too, because there's no data on any of that. So, what's the dose? What's the duration? So that gets to be scary stuff as well. But presumably, if somebody were able to do that, and assuming it were able to clear into the bloodstream, because it is a fairly good-sized peptide, then it certainly could have the potential to tie up and inhibit myostatin in the blood. So anyway,
yeah,
we'll see.
Yeah. All right, Darryn, thank you so much. I think we're out of time, but we have one final thing, and that is give the listeners a take-home message on the talk that we had today.
I would say a take-home message is, is that a lot of times people think that muscle damage is a bad thing. And that being said, is that there are even a number of nutritional supplements that are marketed, and some of them have data to support them, that are marketed to be able to minimize muscle damage. You see it fairly routinely, because and it's based on the premise that muscle damage is associated with muscle protein breakdown, and so if that happens, then you may actually be instigating a reduction in the muscle's ability to undergo an anabolic response, because muscle is being degraded and broken down, and instead of synthesized, but the problem with that is that's not necessarily the case, because we know that muscle damage is necessary for muscle regeneration and for muscle hypertrophy over time with successive bouts of training. It's got to happen. So by providing some type of adjunctive therapy, like a supplement that may mute muscle damage, is actually probably not a wise idea, because you don't want to minimize muscle damage. You want to allow for muscle damage so that you can get a much more robust regenerative response through satellite cell activation. Because if you minimize the damage, then you're going to therefore also minimize the amount of satellite cell activity that's called into play to help regenerate that the muscle that was damaged. So my take-home message is don't be concerned about muscle damage in terms of minimizing it and finding ways to minimize it. Instead, embrace it. Yeah, even all the soreness that might come with it, embrace it, because that is going to provide a very, very, very good catalyst for you to help to ensure that you're going to provide a very effective adaptive response for muscle hypertrophy.
Excellent. This was a great talk, Darryn. I really appreciate you took the time to come back on the podcast. For the listeners out there that want to know more about you or find more information about you, where can they find that?
Probably the best is just on the Baylor website, Baylor, Baylor, Baylor.edu. And then, if have access to PubMed, go in and type in Willoughby DS, and you'll get a list of of my publications and number of things that we've done research-wise in the lab, and a good amount of we talked about today. We've got published papers on, and so that would probably be and those two, and also through the website for the ISSN, International Society of Sport Nutrition. So, which is the sportnutritionsociety.org. So, so those three avenues would probably give you as much information as you might need. I'm on Facebook. You can just type in my name, Darryn, Darryn, Willoughby. And I should come up, and so don't hesitate to send me a friend request, and through there I often post. I don't post a whole lot of personal stuff on there, that site, because it's more business related. So we post stuff about stuff we have going on in the lab and research studies that we have that are newly published, and so on and so forth. And so so that would be the best way.
Awesome. All right, Darryn, thank you so much again for taking the time to be here today. I had a really great time as always, and I need to maybe try and get you more frequently on this podcast so we can go deeper into the rabbit hole talking about muscle physiology.
It sounds great to me. I'd be happy to anytime. Just give me a shout out and say let's do it. So
yeah,
I enjoy it as well. So it's an honor, and it's and a privilege, and I appreciate it very much.
Thank you so much, Darryn. Really, really nice to hear. All right, I'm off to bed. Thanks again for your time, and hopefully we'll speak soon again.
All right, take care, my friend.
You too. Bye-bye.
See ya.